The seventh part in my How Evolution Works series is going to be discussing speciation, which is the natural process by which new species emerge. Over the past 100 or so years, there have been hundreds of documented, observed examples of new species emerging. In fact, it happens so frequently that there are even different types and categories of speciation to describe how this occurs, and I'll be going into each of those types as well as examples of them happening in this video. Before I begin, however, it's important to understand exactly what speciation is. Nobody is suggesting that a dog will give birth to a cat or anything like that. That is simply not how new species emerge. Instead, it's a gradual process, although it can be quicker in certain situations than others. For useful metaphor or analogy to exactly how this can happen, please check out the linked video. It's also useful to begin by defining exactly what a species is. Now, fortunately for biology, there's really not a lot of ambiguity since most biologists use what's called as the biological species concept. That is, two organisms are considered to be the same species if they can produce viable offspring. That is, if they can interbreed and produce offspring, then, well, they're the same species. If they cannot, then they are not. Now, note that this is not necessarily based on exactly how organisms look. One good example of this are the happy face spiders. They look completely different, but they can interbreed and are hence the exact same species. Overall, there are two major evolutionary processes by which new species can be formed. The first is simply general speciation, which I'll discuss in a minute. The second is called polyploidy, which results in a relatively instant creation of new species. Now, because this involves genetic abnormalities, it typically involves plants, and I'll be discussing that at the latter portion of the video. As far as general speciation is concerned, there are four types: allopatric, peripatric, paropatric, and sympatric. For the purposes of this discussion, however, peripatric and parapatric are so similar that we'll simply discuss peripatric. So to begin, allopatric speciation occurs when one large population is separated into two smaller populations by, for example, a barrier. Let's say that there's one large population of birds, and a mountain range pops up in the middle of their population, preventing the birds from flying and mixing with the other side. Over time, because the two populations aren't mixing genes, differences will accumulate and thus lead to new species. Perhaps the most famous example of allopatric speciation occurs in Darwin's finches. Now, when the Galapagos Islands formed, it provided a new habitat for the mainland finches to go on and diversify. Now, these new islands had many different habitats, and the finches could modify and select and fit into each different niche if they modified their beaks. So what do we see is we see the main finch type, but we see differing beaks allowing them to eat different foods. Some of them have large beaks for eating larger seeds, some of them have smaller beaks for eating smaller seeds. Some have beaks that are adapted for eating insects and nectar. Some have beaks like woodpeckers. And this modification is basically occurring through allopatric speciation. And no, they are not able to mate with each other. Another example of allopatric speciation occurs in house mice species on the island of Madeira, which is a Portuguese island off the northwest coast of Africa. These mice demonstrate allopatric speciation because they are physically separated by different valleys leading down to the sea from the mountainous mainland. Now, when one actually examines these mice, you'll find that there are 6 distinct populations, and each of them have a distinct karyotype, or number of chromosomes. These number of chromosome differences resulted from Robertsonian translocations, which are basically errors during reproduction where two chromosomes fuse. This is also the same thing that happened, by the way, in the Ken Miller of Apes and Humans video, which I've linked several times, where the process of the fusion is not necessarily creating the new species, it's merely an artifact of reproductive isolation. Now, this will also prevent the two populations from mixing, hence new species formed. So here you have a clear example of new species being formed. They've got different numbers of chromosomes, can't interbreed, and they came from the same common ancestor on the mountain on the island. The second type of speciation we'll be discussing today is called peripatric speciation. Now, with allopatric speciation, which occurred when, like, say, a mountain range was dividing one large population into two smaller ones, peripatric speciation involves one population and fringe members of it exploiting a new niche and accumulating enough differences to prevent breeding. So the key difference here would be that with allopatric, you have a physical barrier separating the two, whereas with peripatric speciation, there's a new niche. Let's say, for example, that a new type of nut, or a new type of tree with a new type of nut that a certain bird was eating, started to appear on the fringe of a population. Well, certain members of that population with larger beaks could be selected to eat those nuts. Over time, they could become specialized to only eat those nuts, and stop interbreeding with those who do not eat those nuts. Over time, enough genetic differences will accumulate, and boom, you have a new species. There are several documented examples of this happening as well. Another wonderful example of peripatric speciation occurs in the Australian bird Petroica multicolor. Now, I won't really get into the details of the speciation event, however, I did stumble upon a really neat paper while researching this video discussing the molecular phylogeny of it and tracing its ancestrage and lineage through mitochondrial DNA, and I'll link that right now. Another example of peripatric speciation can be found in the London underground mosquito, or Culex pipiens, which basically emerged over the last few decades when the London underground system was built. Basically, overground species of mosquito, or an overground species of mosquito, colonized, went underground, exploited the new niche, and it now is very different from its predecessor. The new species has completely different mating behaviors, it's therefore difficult to mate, and it also shows genetic drift. Here are sources citing that information. The last type of general speciation I'll be discussing is called sympatric speciation, which is where species diverge while they're inhabiting the same place at the same time. One example of sympatric speciation that's occurring actually right now exists in that of hawthorn flies. Now, the hawthorn flies, are also known as the apple maggot flies, are their normal hosts are the hawthorn trees. They're native to North America. But sometime in the early 1800s, they began to infest apple trees. Now, what happens when we take a look at these two different populations, that is, one population infecting hawthorns and the others that are infecting the apple trees? We find that there are significant genetic differences between them. For example, 16 out of the 13 Alzheimer's mellici, if you're interested in the specifics. Also, there are major differences in maturation. The species of apple flies tend to mature at 40 days, where the ones infecting the hawthorn trees mature at 54 to 60 days. So this is the middle, or the beginning rather, of a speciation event that we can actually sit back and take time and watch. It's occurring in nature. We've got species of these beetles, or examples, or specimens rather, of these beetles dating back hundreds of years, so we can see exactly what they look like before, and we can watch the changes unfold. Now, a really interesting example of sympatric speciation can be found in the three-spined stickleback, in which there are two species. One is a larger benthic species that has a large mouth and it feeds on prey, large prey rather. Now, there's also a smaller species which has a smaller mouth and, of course, feeds on plankton. Now, some interesting things about this are that the DNA analysis shows that each lake was independently colonized sometime after the last ice age by a marine ancestor. DNA analysis also shows that the two species in each lake are more closely related to each other than they are to any of the other species in the other lakes. But nevertheless, these two species are reproductively isolated and will not mate with each other. They are separate species. Now, something very interesting happens when we place them in aquariums, and that is that the larger species from one lake will spawn with the larger species of another lake, and the smaller plankton-eating species of another lake will spawn with the smaller plankton-eating species of another lake. But of course they will not mix. The last thing that I wanted to touch on here at the end of the video was discussing what factors favor the accumulation of genetic differences. Because keep in mind, these accumulation of genetic differences between two populations is exactly what drives speciation. There are several things. First of all, a small population size. This greatly enhances speciation as well as genetic drift. A second thing is assortative mating, which occurs within a species, and it's basically the tendency for certain individuals to mate with individuals like themselves. For example, in a species of beetle, the larger individuals of that species will like to typically mate with larger females. Same thing with, smaller males will like to mate with smaller females, and this can greatly enhance and provides a unique opportunity for sympatric speciation. This can also be demonstrated experimentally in the case of flower beetles. In the laboratory, that is. There were researchers in 1981 took a population of flower beetles and divided them off of each generation. They would take the 8 largest males, the 8 largest females, and have them mate, and then they'd take the 8 smallest males and 8 smallest females and have them mate. And they would basically let this go on, the large mating with the large, and the females, and the small rather mating only with the small. And they continued this for 15 generations. At the end of it, it was demonstrated that assortative mating had occurred in half of the experimental lines. A researcher named Diane Dodd also repeated this experiment, only using Drosophila or fruit flies as her agent. What she did is she took one large population of fruit flies and arbitrarily divided them up, fed half of them only maltose food, and the other half only starch food. And now these starch versus maltose subpopulations were kept separate and allowed to breed, and many generations were able to pass. What she found is when she put them back together, those raised on maltose food would not mate again with those eating only starch food, even though they came from the same population to begin with. So this is the formation at the beginning of a mating barrier, which would perfectly cause speciation because, again, it's preventing genes from flowing back and forth. Lastly, a logical question arises, and that is, if these two populations are separate, what's to prevent them from coming back together? And the answer is isolation mechanisms. An example of this would be birds having complex dances. If the other bird is a member of a different species, it won't know the dance and it won't mate with it. This is also seen in Galapagos finches, who select their mates based on different beak sizes. Different species have different sized beaks, and thus they won't mate. Another thing is that this is often seen in insects, is that the sex organs physically and structurally are not compatible. Another thing that could happen is that the sperm could fail to reach the egg. These are termed prezygotic and postzygotic mechanisms, or isolation mechanisms, and it's actually a fairly good read if you have the time.